everyone, and welcome back to the Smart Tea podcast, where we talk about the lives of scientists and innovators who shape the world. I'm Aarati.
And I'm Jyoti, her mom.
Mom, I don't think a lot of people are too familiar with you yet. You're still fairly new to the podcast. But one thing that maybe people don't know is that you did your master's degree in microbiology, correct?
Yes, I completed my master's in microbiology in India. And
Mm-hmm.
then I came to the US with my husband, Artist dad. Um, and then I worked in various academic institutions, and eventually I started working in biotech companies in the Bay Area, where I have acquired a lot of skills like molecular biology, protein chemistry, cell biology. Basically, I'm a biotechie at this point.
But your very original classes that you
Yeah,
took.
my roots are rooted in microbiology, so to
Yeah.
speak. Haha.
Any particular reason why you chose microbiology as your field?
I initially thought that I wanted to be a doctor, like in
Mm-hmm.
practicing medicine. I had all these beautiful visions of myself being part of Doctors Without Borders, and
Oh,
lots of
wow.
lots of noble thoughts there. However, the field is extremely competitive in India. It was very even now it is, but I did not get admission into a medical school, and
Mm-hmm.
so I was heartbroken. Really, literally, I was heartbroken. I did not know what to choose, and then
Mm-hmm.
talking to other doctors, talking to many people around me, they said to me, microbiology will allow you to become more like a pathologist. You can still be on the clinical side of things. So I
Yeah.
thought, yeah, that sounds right. I would still be helping people. I would still be fighting diseases. I would still be bringing medicine to the people in one way or the other, maybe not as the primary doctor, but something else. So that
Yes.
is how I went into microbiology.
Oh, nice. That is a lot more noble and a much better reason than why I did biochemistry as my major, which was simply because my dad had done it. So,
No, but you were inspired by people close to you. That is a good thought as well.
yes. So, anyway, the reason I was asking about your roots in microbiology is because today we will be talking about an extreme microbiologist. His
Oh,
name
nice.
is Thomas Brock. I don't
Okay.
know if you've heard of him.
I don't think I've heard the name.
Well, you've definitely heard of the microbe that he is most famous for discovering, which
Okay.
is Thermus aquaticus or TAC.
Of
Of
course,
course, we know.
legendary microbiome that is oh my goodness,
Yeah.
changed the entire field. You can call Thermus aquaticus as the mother of biotech, basically, or
Yeah.
the organism that gave birth to the biotech industry. I would not have a job without Thermus aquaticus today.
Or you would have a job, but it would be very tedious, as we will get into. So
Yes. Oh, this is going to be such a fun story.
it's a really good story. So I'm excited to tell you about him and what led him to his discovery.
Nice.
Thomas Dale Brock went by Tom, and he was born in Cleveland, Ohio on September 10th, 1926. He
Mhm.
was the only child to his parents. His father was also named Thomas, and his mother was named Helen.
Okay.
So, obviously, since he's named after his father, as we go through his childhood, I'm going to be calling him Tom and
Okay.
his father Thomas, just to keep them straight. So, Cleveland at this time was a major industrial city in the 1920s, but Tom's home was far away from the main city, sitting
Mm.
on top of a hill where they had a distant view of Lake Erie and they had fields of cows and woods all around them. So, he's not like in the city proper. He said, quote, I did have a stable home life and a pleasant neighborhood with lots of friendly playmates.
Sounds idyllic. Idyllic,
Yes.
idyllic childhood.
So Thomas, his father, worked as an electrical engineer, and his mother was a registered nurse, but for most of his childhood, his mom was a stay at home parent.
Okay.
So they were very much part of the working class. And then once the Great Depression hit, they
Yeah.
only managed to get by because Thomas was luckily able to hold on to his job at the boiler room of St. Luke's Hospital.
Okay.
So they're getting by, but by no means do they have a ton of money to spend.
Yes. Hmm.
But Thomas did recognize that part of the reason his career potential was limited was because he didn't have a formal education.
Hmm.
So he couldn't climb very high up the corporate ladder.
Hmm.
So he encouraged his son, young Tom, to pursue an education. He would bring home discarded electrical equipment and show Tom how to build radios and electromagnets. And when he was 10 years old, he got a chemistry set as a Christmas present. And he and his dad set up a little laboratory in the basement and would
Nice.
do little
That
experiments together.
that is so nice. It sounds like the dad did everything he could to foster a love for learning in his son. That
Yes.
that kind of good encouragement. I like to hear stories like that.
But unfortunately, when Tom was 14 years old, his father became extremely ill and passed away.
So sad. Hmm.
Yeah. So this left Tom and his mother basically in poverty. So
Yes.
they moved back to the mother's hometown of, oh my God, I looked this up, Chillicothe, Ohio. I believe
Okay.
that's how you say it.
Okay.
Chillicothe, Ohio.
Hopefully somebody will correct us if
Yes,
we are wrong.
pretty
All
sure that's how
right.
you say it.
Okay.
So they moved back to his mother's hometown of Chillicothe, Ohio.
Mm-hmm.
His mom was able to get a job as a nurse, but Tom had to help out too by getting jobs at the grocery stores, drugstores, like just little jobs here and there. They didn't pay much, but right now we're in World War II, so there's a lot of labor shortages in the
Yes.
US. And so
Yes.
Tom never really had a problem finding little
Finding a
odd
job.
jobs to do.
Yes.
He also continued his interest in science, now with a friend named David Thornburg, who
Mm-hmm.
was also interested in chemistry.
Okay.
So, a lot of the information, I should say, for this episode came straight from an article that Tom Brock wrote about his own life, a short autobiography, like a 30
Yes.
page article. But he said very nonchalantly that he and David set up a small laboratory in the loft of a barn behind Tom's house where they, quote, did a lot of crazy experiments, parentheses, explosives, toxic gas, and parentheses.
Oh my God.
I was like,
This is
just,
just a couple of boys dinking
yeah,
around
yeah,
with little.
just,
We'll play with a little bit of gunpowder today, and
yeah,
tomorrow we'll play with that gas and see if it is poisonous after all.
it's fine. I got my hand on some nitrous and some sulfuric acid. Let's just see what happens.
But if you think about it in retrospect, that kind of childhood is really fun. I imagine it to be a lot of fun. I was pretty nerdy and straight laced myself, so I have to just
Hmm.
imagine this kind of really fun childhood. But still, this sounds like a good
Yeah,
amount of fun.
it's very nostalgic to even think about. Just like
Yes,
two boys in a barn loft
yes.
having fun, messing around. I have to say, I was quite disappointed that this was such a throwaway one line in his autobiography. I was like, what? What?
Tell
Tell
me
me
more.
more. Tell me
Tell me
more.
more. Tell me more.
Yeah. Yes. So, also, his friend David Thornburg had heard about penicillin.
Yes, Alexander Fleming. Alexander Fleming discovered penicillin.
Yes.
That's a very interesting story as well, actually.
I know. I kind of want to cover him at some point on the podcast,
Okay,
but
so
I
I
think
won't go
his
into that
no,
at
no, but
this point.
his story is famous, so you can.
Yeah, the way he discovered penicillin was simply fantastic. So he was working on something else entirely,
Mhm.
and he was having bacteria on little plates. You have agar plates on which you grow bacteria, and he was a messy guy. Like, he did not clean up the plates from his experiment on a daily basis. He
Mhm.
would leave them around for weeks at a time. And after some time, he noticed that there was this ring of area on the plate where the bacteria were not growing.
Yeah.
And in the center of the ring was this mold
Like this
that was
moldy spore.
the
Yeah.
mold that was making penicillin. And that is
Yeah.
how he was very smart, obviously, and he connected the dots.
put it together that there's this mold that is obviously producing this chemical that is killing the
That is preventing
bacteria.
the bacteria, and he saw
Yeah.
the implications immediately in terms of how we can use that for
Hmm.
ourselves. And the story, that what I was trying to remember was that later on he, of course, he became very famous, etc., etc., and he was offered a brilliant, beautiful new lab, all spick and span and clean. And his
Hmm.
first comment was that he said, "Oh my God, if I had been working in such a beautiful, clean environment, I would never have discovered penicillin."
Yeah.
Yes, sorry, I took this off on a big
No, but
tangent, but
No, but
um,
this exact story, I think, is what really captured the imagination of young Tom and his friend David. And they also were like, this guy has made such a great discovery from some mold growing on some plates. Maybe we also can do something to our soil and enrich it to grow microorganisms that will produce antibiotics, like right here in our own backyard. And so
so
they
that kind of a
also,
story was such an inspiration. Some boys dream of killing dragons and being a hero, but they were dreaming of finding molds
yeah,
that could
find the
that could
next
make
big
find the
penicillin.
next
We'll
big
probably get
penicillin.
rich.
Yeah,
Yeah.
or even save mankind. I
Yeah.
mean, penicillin
That
was game
too.
changing for
That too.
mankind too.
Yes. What does it say about me that I was like, get rich? Yeah. So after high school, Tom really wanted to go to college and study chemistry, but due to the ongoing war and their financial constraints, he couldn't really pay for tuition. So he
Hmm.
decided to enlist in the Navy and get an education in electronics through their educational programs,
Okay.
which makes sense. His father taught
Yes,
him about electronics and,
all those broken radios that his father brought home to.
yeah. But it turns out the Navy was a huge culture shock for him after his
Hmm.
quiet, idyllic childhood.
Yeah.
He spent about 18 months going to Navy schools in Chicago. And for the final few months, he was sent to Kodiak, Alaska as
Hmm.
part of the shore patrol, where his job was to make sure that when curfew came around, none of the soldiers were hanging around in bars or brothels.
Oh
So he had to go
wow.
kick them out of,
Oh my God, that's a hard one. It's almost like being a class monitor and being the person who gets to spoil everybody else's fun. That's not a good position to be in.
and especially after his like quiet childhood where he's like
Yes.
playing in,
Yeah.
the barn and
The
the farm and everything. And he's
yeah,
like,
the
oh,
tedious enforcer
yeah,
of law. Oh.
yeah. But the Navy also gave him access to a ton of books. And so he was
Hmm.
able to start reading all of this great literature that he had never
Hmm.
had access to before.
Hmm. Hmm.
And by the time his Navy career was over, he had changed his mind and he decided he wanted to become a writer.
Oh wow.
By 1946, the war is over, and now
Hmm.
Tom is considered to be a military veteran. So he's able to take advantage of the GI Bill, which
Awesome.
provided education and other benefits to
Yeah.
veterans.
Yeah.
So he goes back to Ohio and he enrolls at Ohio State University to study
Hmm.
literature and writing.
Oh, okay. Hmm.
Hmm. But very soon he changes his mind again.
Okay.
He says that for one of his literature courses, he had to read a book called Aerosmith by Sinclair Lewis. So
Ah.
it's Arrow as an Arrow, not Aerosmith like the band. I
Oh, I
know.
did think of Aerosmith the band. Okay,
Did you?
I was getting, yes, I did actually, and I was getting very confused. I'm like, that sounds like it must have been after his time. How did that happen?
Yeah, I also did some research to see if Aerosmith, the band, got their name from this book, but apparently not. But the protagonist in the book is a bacteriologist, and he says that's probably what pushed him back towards science. So he goes back to biology and he ends up finishing a degree in botany, of all
Okay,
things.
okay,
He was then offered a graduate position also at Ohio State University,
mm-hmm,
but
mm-hmm.
he soon got bored of studying plants.
Okay,
So he switched again to mycology, which is the study of fungi, which
yes.
again
Again,
makes a
it's
lot of
going back
sense.
to that
Yeah.
same thought process. Yeah, I want to be the
Yes.
next Alexander Fleming. Yes,
Yes.
yes.
He ended up doing both his master's and PhD in mycology. He also got married towards the end of his PhD to another scientist, Mary Louise Loudon, who had her master's in microbiology.
Okay,
After his PhD, he was having a tough time finding a job in academia. He
Hmm.
got a temporary position as a research associate at the Ohio Agricultural Experiment Station working on soil fungi, and he was able to pivot that into a full-time job at the Upjohn Company in their antibiotics research department in
Hmm.
Kalamazoo, Michigan.
Yes. So he is coming to where he wants to be.
Kind
With
of, yes.
yeah, getting there. He's getting there.
He's getting there. So, this
Yeah.
is where he was truly introduced to bacteria and the wider world of microbes. He spent 5 years there studying the relationship between microbes and antibiotics, and he was publishing papers. And he also found that because he was working for a company, it was an 8 to 5 job. And so he had a lot of free time. So he started exploring his local area. He found an abandoned railroad that captured his interest.
Hmm.
So he did a deep dive into the local history of that railroad and the surrounding area and published articles about it. So he's getting his like writing bug
Yes.
out too because he
That's
had
right.
wanted to
That's
be an
right.
author.
He's a man of many interests. He would have been such a fun person to talk to and get to know.
Hmm.
His neurons are firing all the time. It's like
Yeah.
anything you tell sparks his interest, and he's off chasing that.
Yeah, it definitely seemed like that.
Yeah, they're brimming with life and vitality, these kind of
Yeah.
people. Yeah,
Yeah.
they have so many good ideas. It's like
Yes.
one lifetime is not enough. I need to do this and this and this and this, and time is too
Yeah.
short.
Speaking of exactly that, he also started teaching himself German and he got really
Mm.
good at it and was even able to translate scientific papers about microbiology from German into English.
Okay, out of this
Yeah.
world.
But as seems to be kind of a running theme
Hmm.
here in his life, he soon got bored of this. The corporate world was very routine, and he wasn't on the cutting edge of science and discovery at all, and he had no control over what he got to study. So he quit his job and moved back to Cleveland, determined to get back into academia. But this was really tough because he had been out of the academic world for like 5 years now.
Okay,
But finally, he got a position as an assistant professor at the Western Reserve University's biology department.
okay.
The pay was about half of what he got at the Upjohn Company, but he was much happier because he was teaching microbiology and
Yes,
mycology and
yes,
genetics.
yes,
And while he's teaching, they say teaching is the best way to learn, right? So he's
yes,
also learning about the subjects that he's teaching
yes,
in the
yes.
process. He also was able to pursue some research projects of his own, and from that, he was able to publish 13 papers related
Hmm,
to the
hmm,
mode of action of antibiotics and yeast mating.
hmm.
He even wrote a book called Milestones in Microbiology that largely was helped by him being able to translate German papers because a lot of early work in the field was deeply tied to Robert Koch, who was a German,
Yes, that's right.
scientist.
That's right. Tuberculosis,
Yes,
right? Isn't he the
tuberculosis,
tuberculosis guy?
yes,
Yes.
cholera,
Yeah.
anthrax.
Yes.
Yeah,
But that is again fantastic communication. Like he's
yes,
being a science communicator again, because
yes, I love
he's
it.
yes, he's bringing science from another language to
Mm-hmm.
the scientists in America. And
Yes,
now there is a transfer of
yeah,
information, which is always creates synergy.
yes. So I loved that he did that. Um, the academic world was also a lot more flexible in terms of days off and vacation time. So he spent many weeks with Louise's father in northern Ontario and developed a great appreciation for exploring the outdoors. He especially loved anything to do with spending time near water. So
Yes.
fishing, boating,
Hmm.
kayaking, all of that. And he felt that as an academic, now he finally had the freedom to study things that caught his attention while he was out in nature. So he could see something and be like, "Ooh, what
Yes.
is that?"
Yes. Yes.
Go
Yes.
and figure it out.
Yes.
In his second year at WRU, the chairman of the Department of Microbiology, Dr. Lester O. Krampitz, offered him a postdoc position in his lab. While I was reading this, I was like, oh my gosh, it almost seems like he's going a step back. Like he graduated, he got a job, he had an assistant professorship, and now he's going
Yeah.
back to doing a postdoc.
but maybe it looks backwards, but maybe that's the right play path. Let us not
Yeah.
let's
No, it
let's hold
was.
judgment. Let us hold
Yeah.
judgment.
Well, he
Yeah,
certainly thought it was the right path because he was like,
absolutely. Yeah, he is smart. I trust his judgments.
yes. So he resigned his job as an assistant professor because he wanted to be able to do more research and
Yes.
less lecturing. After a year there as a postdoc, he finally felt like he could legitimately call himself a microbiologist. And
Huh.
so now, in 1960, 8 years after getting his PhD, he was finally ready to settle down, quote unquote.
Okay. Yes.
Yes,
Yes.
he's like
Yes.
gone from electrician to writer to botanist to
Yes.
finally,
Mycologist
yeah,
and then teaching here and there.
yeah,
But that kind of interdisciplinary knowledge is extremely valuable.
yes. So, a position for an assistant professor of bacteriology opened up at Indiana University in Bloomington, which
Hmm.
was a small university town. And that kind of life seemed like a great change for him from the
Hmm.
city life of Cleveland.
His childhood was like that, very
Yeah.
idyllic and
And I think that's what he was
mhm.
kind of going back to because Cleveland is this big city. And so
Yeah,
he's like, yeah, let's go back to something a little more charming, a little bit more
yeah, a
like
bit more
slow
rural.
pace. Yeah.
Yeah, yeah. Get rid of this hustle and bustle life. Yeah.
Yeah. So Tom and Louise make the move to Indiana, where his
Hmm.
focus now became medical microbiology because
Hmm.
that's the position he took basically.
Yes.
But Tom never really could let go of his background in ecology, especially now because after spending so much time with his father in law out on the lakes of Ontario, he had become very outdoorsy.
Hmm.
And so now in 1962, we get to a big turning point in his career.
Okay.
He travels to San Francisco to attend a conference for the American Society for Cell Biology.
Okay.
And during his time here, he visits the Hopkins Marine Station and
Okay.
sees the lab research going on there. And it's the first time that he's really seen a marine biology research lab. And he becomes absolutely fascinated by
Oh
this.
wow. Okay.
And pretty much immediately, he starts making plans to kind of do his own research the following
Yes.
summer at the marine lab.
Okay. It just caught him.
Yeah.
It just caught his fancy.
Yeah, so a visiting scientist from the University of Washington told him about a research facility called Friday Harbor Laboratories.
Hmm.
So
Hmm.
he contacted the director and said he wanted to spend the following summer of 1963
Hmm.
there to
Hmm.
study the presence of certain types of bacteria in marine animals.
Bacteria in animals. Okay.
Yeah,
Okay. Yeah.
he
Okay.
wants to study microbiology, like marine microbiology
Yes.
basically.
Yes.
Yeah.
Yes. Hmm.
So he does that. He goes there in the summer of 63 and he
Yes.
studies a microbe called Leucothrix mucor, which
Okay.
is a long filamentous bacteria that is
Hmm.
found in a lot of marine species and can cause disease and impact animal development.
Okay.
But the really cool thing that Tom observed was that when he grew these bacteria, Leucothrix mucor, in the lab, when the bacterial culture became overcrowded, the bacteria would literally tie itself into knots.
Wow.
And over time, the knots became tighter and tighter until the cells that were in the knot would fuse together and they would make a bulb that would break off into two filaments.
Oh
And this
wow.
was a new way that the bacteria was able to reproduce, essentially.
Oh, it's a new propagatory mechanism. And so the more constricted its space, it
Hmm.
it leads to more and more this tightening, and then it tightens itself into a ball. And
So I think it was like if you imagine like a piece of string and you tie a
yes,
knot in the middle of the string, that
yes,
knot in the center of the string would become so tight that it would break. And now you have two bacteria instead of the
yes,
one long
yes.
one. This was a new way that the bacteria was able to reproduce that had never been observed before.
Okay.
So he had made all these sketches to show what he was seeing. And that figure from his paper made it onto the cover of Science magazine and was also featured in the New York Times because it's
Oh
such a cool thing.
wow. Yes.
I mean, for me, that would be my crowning achievement. I would be like, look at this thing that I discovered. Heck yeah.
Absolutely, and I'm featured in one of the two of the most famous magazines of the world. That's it.
Yeah.
I'm done. I'm done. I can retire
I'd
now.
be like,
I'm
I'm done.
retired. Yes,
Yeah.
yes,
But for him, this just blew open the field of microbiology for him
yes,
because he was like, if the world didn't know about this behavior of Leucothorax mucor tying itself
hmm
into knots, which is so cool, what else is out there waiting to be discovered?
yes. The possibilities are endless.
Yeah, we know so little about microbiology.
It takes a genius to understand that. Also, it takes a
Yes.
genius to understand how ignorant we are. It's only the very foolish who think that we know everything.
Yes. So his work on Leucothrix mucor got him interested in another filamentous bacteria called Theothrix,
Okay.
which grows in sulfur springs, which is a very unique environment for anything to be growing.
Yes, an extreme.
Yes.
So that's where when you introduced him, you said something about extreme bacteria, and this is what
Yes,
he's
he's an
doing.
extreme
He's
microbiologist.
an extreme microbiologist. I get
Yeah.
it. I get it. So he's looking at things that grow in extremely weird situations.
Yeah, because Leucothrix mucor only exhibited this behavior when it was extremely overcrowded. If the culture was kept at sort of a normal concentration,
Yeah.
everything was fine and they just reproduced however they normally reproduced. But
Yes.
if you let them overgrow and you let them like get crowded, then they started exhibiting this different way of reproducing.
Yes.
And so that's what he's like, oh, okay, they get extremely crowded, they behave in a different way. Then he learns about Theothrix, which can survive in this extreme condition of having high concentrations
Yes.
of sulfur. And the
Yes.
concentration of those minerals is normally prohibitive to
Mm,
growth. And
yes.
this bacteria can survive there.
Correct.
So he was really excited by this and how much there was to learn and discover. And so he starts planning another book called Principles of Microbial Biology
Mm.
that would go into this unknown world of microbes in nature.
Yeah.
And so, in the name of research, he starts traveling around trying to get his hands on microbes that live in these kind of unique
Yeah.
ecosystems.
Niche environments.
So that Christmas, he took a solo trip to South Florida,
Mm.
driving a VW minibus that he and
Mm.
his wife had converted into a camper van.
Ah, sounds like again. He is a fun guy to be around. Look at
Yeah.
the kind of fun ideas he has.
And in Florida, he visited a large sulfur spring in the Caribbean gardens,
Mm.
and he got his hands on some Theothrix for the first time. Then the following summer, he headed back to Friday Harbor Laboratories, which
Hmm.
was in Seattle, Washington.
Okay.
But he and Louise made kind of a road trip out of it. So they
Yes.
first stopped at Grand Teton National Park for a
Aha.
backpacking trip,
Yes.
and then they went on to Yellowstone Park.
Yes.
So, funnily enough, he hadn't really been interested in going to Yellowstone because he thought it was more of like a tourist attraction rather
Hmm.
than a place where you could kind of discover nature or be at peace or observe anything of interest.
Yes.
But, there are hot springs there, so he was like, I should go and
Sure.
see
Let's
what
do
I
it.
can
Yeah.
see.
Uh
Yeah.
huh.
When he got there, he was completely blown away. In
Yes.
his book called A Scientist in Yellowstone Park, he wrote, quote, I was stunned by the colors and the abundance of phototropes in the thermal waters flowing
Nice.
across the center into the lake, end
Mm.
quote.
Nice. So the Yellowstone had become such a big attraction that he's like, nah,
Yeah,
not for a
no,
person like me.
I don't want to.
I don't do things like that. I don't do touristy things.
And he probably thought, like, oh, it's not natural anymore.
Yeah, they must have trampled all over nature. There's nothing
Yeah,
good to see anymore. And then he
yes,
goes there and he's just
just
in
blown away.
stunned by
Yeah,
the beauty.
stunned.
That's beautiful. Beautiful.
So he spent the summer at Friday Harbor Labs again, and then on the way back to Indiana, he stopped by Yellowstone again to collect some
Yes.
samples from the sulfur
Mm,
rich springs of the park.
yes.
And specifically, he was looking for that Theothrix bacteria.
Yes.
He didn't get any because
Okay.
what he didn't realize at the time was that the geysers at Yellowstone were too hot and acidic for Theothrix bacteria to survive there.
Yes.
But despite this, he's totally hooked now. He's like, Yellowstone, that's where
This
I've got
is,
to be.
yes, this is the best place on earth. Yes.
Yes. So the next summer in 1965, he and Louise go back to Yellowstone, this
Hmm.
time bringing with them nutrient rich media to grow the bacteria that they collected.
Ah,
Yeah.
smart. Here they're
Yes.
pre-planning it now. They're like,
Yeah.
we are not getting anything because by the time we get back all the way to Indiana, things are not working out. We'll
Yeah.
make sure that we grow them correctly right away.
Yeah.
Yes,
And I just
yes,
love this
yes,
idea too of them like packing their suitcases. Like, did you get your
yes,
socks? Did you get your
yeah,
shirt? Did you get the
yeah,
nutrient rich
yes.
media for the
Media,
bacteria?
and
Yeah.
it's great that he is married to a microbiologist as well.
Mm-hmm.
So the
Yes.
both of them are on the same page. If he forgets his nutrient rich broth, then
Yes.
she's going to remind
She's
him.
got it. Yeah. She's
She's
got
got
it.
it. Yeah. Yes. Fabulous.
So on this trip, they discovered huge masses of pink, stringy material growing
Hmm.
in a hot spring channel. And this
Ah.
caught their interest because most of the time, scientists were studying bacteria that either grow in the soil or in our bodies.
Yes.
And normally that means they're growing in a temperature range of 20 to 45 degrees Celsius.
Hmm.
And they knew, like scientists knew that there were thermophilic bacteria that could grow at a little bit of higher temperatures. They could grow at 55 to 60 degrees Celsius, but they thought that that was kind of the limit for microbes, or at least they hadn't really looked any higher.
Yes.
But there's this pink stuff that's growing in this hot spring channel, and the water there is like 80 degrees
Ooh.
Celsius.
Yes.
And when he asked the park rangers what they thought it was, they said they guessed it was algae. But
Uh-huh.
when Tom and Louise looked at its composition, they found that it had no chlorophyll, like algae would have had, and it
Yes.
had a very high protein content and a very similar microscopic structure to filamentous bacteria that Tom was
Yes.
familiar with.
Yes.
So, despite there being no record of bacteria being able to grow at such high temperature, he becomes convinced that this pink stringy stuff is bacteria. And this is the first time he starts to think about the idea that maybe extreme thermophiles or hyperthermophiles could exist.
Yes.
So, based on this preliminary research, Tom wrote and received grant funding from the NSF to seriously work on the microbial ecology of Yellowstone.
Oh, nice.
So, they
Yes.
set up a temporary lab in a
Hmm.
rented cabin and they
Oh,
delved deeper into
wow.
this research. Yeah.
That's fabulous.
Yeah. It's like such great
So
times.
attractive.
It's like,
Yeah.
yeah, I want to
That-
do research in the 60s. This
yes,
sounds
I'd like
great.
to do this. I'd like to do this kind of work.
Yeah.
Be in the middle of Yellowstone researching these things. Yeah.
Yeah. Surrounded by
Nice.
gorgeous nature.
Yes. Anytime you want a little tea break, you go out and get beauty surrounding you. Mm,
It sounds amazing.
nice.
I knew you would
Nice.
like this
Nice.
story. I knew you
Yes.
would love this
Yes. It
story.
appeals to everything. It appeals
Yeah.
to the nature lover in me. It appeals to the microbiologist in me.
Yeah.
Yes.
So they set up this temporary lab in a rented cabin and they start publishing papers. The scientific community starts to sit up and take notice of this.
Hmm. Hmm.
And not just the microbiologists and the ecologists, but what he hadn't realized is that when he started this research, the biochemists and the molecular biologists and all these other scientists with different agendas were all reading his papers and asking him questions about like these microbes metabolisms and how they worked and how their
Oh.
survival mechanisms were different.
Yes, the wide ranging implications of a living organism being able to conduct all the processes required for life
Yes.
at a temperature that is beyond what we know as
Mm-hmm.
being able to support life. And so the implications for all processes, biochemical, protein metabolism, everything, all these processes are now impacted.
So
Yeah,
everyone is
how does
just
it
like,
do that
wait,
at
how
that
does it do
temperature? Wait a
this?
second. What does
Yeah.
that mean for my research?
Yeah. If
Yes.
it can survive in this really weird environment, what does that mean for medicine and
Yes.
like cancer and all these diseases that
Everything.
cause
Yeah.
really like our metabolism to go out of whack and,
Nice. Nice.
So Since this work was really starting to take off, they set up a more permanent lab in West
Ah,
Yellowstone.
okay.
And then they were able to start bringing in grad students and lab techs to come and help with this work. In the summer of 1966, a young undergraduate student named Hudson Freeze
Hmm.
came to work on the Yellowstone project.
Okay.
The irony of that last name did not escape me, by the way.
Yeah.
Hudson Freeze, studying extreme
Well,
thermophiles. So Tom gave him the project of trying to culture that pink filamentous organism. So up
mm,
until now, they had been collecting samples constantly to study it, but they hadn't been able to get the formula down for how to keep it growing in the
mm,
lab.
yes.
So Hudson Freeze started trying to figure out what temperature it needed to grow at and what
Mm,
nutrients it needed and how much and
yes.
stuff like
Yeah.
that.
Yeah. Just best growth conditions.
Yeah.
Mhm.
So he wasn't successful, but he did in this process end up sort of accidentally culturing another yellowish bacteria that came from the same samples. And
Oh,
they
so it's
hadn't
a
really
mixed,
noticed it.
mixed. Yes, because the pink guy is so strong
Yes.
that maybe the pink one is growing faster and
Yeah.
taking over. If it's a mixed culture of yellow and pink bacteria,
Yes.
this happens all the time in microbiology. You have to be very careful.
Yeah. So he's not able to get that pink bacteria to grow properly, but whatever he's doing is allowing the yellow bacteria to grow properly.
Mm,
And so they start culturing that one. And
mhm.
Tom called this yellow bacteria Thermus aquaticus or
Oh,
TAC, T A
this
Q.
is the Thermus aquaticus. Okay,
Yes,
okay. Oh my
this is the
God.
one, this yellowish one
Wow.
that came.
When you say the color yellow, it makes me think of gold, and it turned out
Mm,
to be
yes,
gold. It
pure
turned out
gold.
to be pure gold for biotech.
Yes. So all the biochemists and molecular biologists also who are keeping tabs on Tom's work are super excited by Thermus
Yes,
aquaticus.
yes.
Hey, Mom, did you know that Galactic Polymath Education Studio is holding the first ever My Sci Journey competition?
Yes, it's so exciting. I heard that you could win up to $500 for sharing your science story.
Yep. Just make a short video describing your scientific career path and the importance of your research and upload it for your chance to win.
Eligible videos will also become part of an online resource to show the public a glimpse of the real people and work done in science.
The My Sci Journey competition is open to everyone, from high schoolers to tenured college professors, and is a great way to flex your science storytelling muscles.
Upload your submissions by September 30th, 2025. For more competition rules and resources, including beautiful slide templates to help you get started, visit galacticpolymath.com. galacticpolymath.com.
That's galacticpolymath.com.
We
We can't
can't
wait
wait
to
to
hear
hear
your
your
stories.
stories.
Hi everyone, Aarati here. I hope you're enjoying the podcast. If so, and you wish someone would tell your science story, I founded a science communications company called SciComm, that's SYKOM, that can help. SciComm blends creativity with scientific accuracy to create all types of science communications content, including explainer videos, slide presentations, science writing, and more. We work with academic researchers, tech companies, nonprofits, or really any scientist to help simplify your science. Check us out at scikommer.com. That's SYKOMMER.com. Okay, back to the story. We're going to take a little detour here, as I love doing in my stories. So, 7 years later, in 1976, a master's student named Alice Chien at the University of Cincinnati published a paper called Deoxyribonucleic acid polymerase from the
Mhm.
extreme thermophile Thermus aquaticus.
Okay. So he's not Brock is not the one who actually pulled out the
No.
enzyme that I'm very familiar with as a biotechie.
Yeah.
Alice Chien is the one who did that. Okay.
Yes. So
Okay.
this is a landmark paper because DNA polymerases are the enzymes that help create new strands of DNA. So whenever
Yes.
there's a break in the DNA that needs to be fixed, or when a cell is dividing and the DNA needs to be copied, the DNA polymerase is the enzyme that actually builds these new strands.
Yes.
So, super important that she got this. I think at the time, maybe people were like, whatever, but like
Yeah.
it turns out to be a really important
It turns
discovery.
out to be the foundation for big techniques that are used in biotech.
Yeah. And we're about to get into that.
Okay.
So, 9 years later, in
Hmm.
1985, a chemist at the Cetus Corporation in California named Carrie Mullis developed PCR, or the polymerase chain reaction.
Based
Yes.
on the DNA polymerase from Thermus aquaticus.
No, not yet. Not yet,
Not yet.
actually. No.
Oh, wait.
No.
Those two pieces haven't
Those
come
two
together
things
yet.
have not.
Got
No.
it.
So he's developed this
The
thing called
PCR.
PCR, and it's a way to, basically guide the DNA polymerase to make tons of copies of a specific
Yes.
gene or section of DNA. And I know you've done this like a ton of times. How many times do you
Millions
think
of.
you've done this?
I have no idea. If I had $1 for every time I did it, I'd be very rich. But
Yeah.
it is one of the fundamental techniques that are used in biotech industries.
Mm-hmm.
So basically, you're able to just take a piece of DNA and replicate it to as much as you want. Make more of the DNA piece of your choice. But it has three pieces to it, because you have your template DNA that, hey, this is the piece of DNA I want. This is the strand of DNA that I want. But the DNA is double stranded. So the first step is where you have to open it up. You have to heat the DNA,
Yeah, you denature it and kind of break the bonds that are
break
holding
the
the two
bonds that
strands
keep
together.
the two strands together, make each one into a single strand. And
Mm-hmm.
now each of those single strands can become your template, and you figure out what the ends are of this piece that you want to replicate over and over, and you design little pieces of DNA, small, like 18 or 20 nucleotides in each of them.
Mm-hmm.
And these are called primers. They serve as little seeds, almost
Yeah.
like the starting seed. And so the second step is where you lower the temperature. So you have first heated it up very high,
Mm-hmm.
and in order to separate the two pieces of DNA, and then you lower the temperature so that these little seed pieces of DNA, these primers, can attach. And then you kind of increase the temperature slightly so that the seed pieces can then be
Built upon.
built upon. Yeah, they can build upon, and it's called extension. And they
Mm-hmm.
extend and they meet each other and they make a new piece of DNA.
And so you do this with like one piece of DNA, it becomes two pieces.
Yeah,
Yeah.
so one round of PCR makes two pieces of DNA. Then you do this one more time, and you have a machine that does this.
Mm-hmm.
Machine just automatically cycles through these three things, denature, for like separate the pieces, and
Mm-hmm.
then you put the primers on, and then you extend it for 4 or 5 minutes. One more time. That's now you two became four. Do it again. Four becomes eight, and so on and so forth.
So, yeah, this allows you to basically create millions of copies of your gene of interest or your section of DNA that you want to study. And it is such a fundamental tool of biology nowadays that Carrie Mullis actually ended up sharing the Nobel Prize in 1993 for developing PCR. The problem with PCR, the way that Carrie Mullis had developed it originally, was that the scientists, Carrie Mullis and the scientists at the Cetus Corporation, were using a DNA polymerase from E. coli bacteria that
Yeah.
lives in our gut at around
Yes.
37 degrees Celsius all the
Yes.
time.
Yes.
So, in that initial denaturing phase of the PCR cycle, where you have to heat everything up so that the DNA falls apart, the E. coli polymerase would also unravel and stop working.
Yes.
So, after every single cycle, the scientists would have to manually go in and add more polymerase.
Yes.
And that got old really fast. People
Yes,
were
because you
like,
literally
yeah.
are standing there.
Yeah, like every 7 or 8 minutes or something, you have to go
Yes.
and do it again. And you have to do that for like, what, 2, 3 hours straight,
Yes.
just every
Yes.
7 minutes.
Yes. You just
Yeah.
sit there with a stopwatch, and three hours of your life is can be lost like that.
I mean, settle in with a good book and a timer. And
Yes,
like, what
exactly.
else can you
And a
do?
whole bunch of enzyme, and
Yeah.
you keep doing it.
So in 1986, one of Carrie Mullis's colleagues, Randall Saiki, thought, hey, what if we use a polymerase from a thermophilic bacteria instead that could maybe survive that denaturing temperature? So
Very
he
smart.
started using the Taq polymerase from the paper that Alice Chien had reported that she had isolated.
That DNA polymerase. And
Yes.
then everything came together.
So in 1988, Randall Saiki, Carrie Mullis, and their colleagues published a paper in the journal Science called Primer Directed Enzymatic Amplification of DNA with a Thermostable DNA Polymerase. And
Hmm.
the rest is, as you know, history.
Absolutely, absolutely. Like you said, once you bring in, you marry this DNA polymerase from Thermus aquaticus, which can go through the cycles of high and low temperatures, add it in, put all your things together, and walk away.
Yeah.
And when you come back 3 hours later, the one copy of DNA that you started with has now become a million copies.
Mm-hmm.
That just becomes mind blowing in terms of its applications. So, for example, forensic science, you get this little piece of DNA from some crime scene. Now you're able to take it, extract it, put it in, and start a PCR reaction, walk away. And now you have plenty of DNA to work with so that you can identify more.
Yeah, you can do a ton of different tests now on that DNA to figure out whose it is and
Yes,
do DNA
yes.
matching and everything.
It's revolutionary for agriculture because the scientists are working all the time to improve our crops and make them more resistant to disease and less dependent on pesticides and more resistant to extremes of weather. DNA fingerprinting, then medical diagnostics. It
Yes.
is
Huge.
huge in medical diagnostics. So clinical diagnostics, it's just incredible. And then of course we have the cloning, making more of the DNA that we want. We want to create a particular clone that creates this protein. You do use PCR and you make that happen. And you and I, both of us in our scientific careers, have done a lot of that, where we want a
Yes.
particular mutation, we want a particular piece of gene, and there it is.
Yeah,
You have
because
been able to
literally
make it.
every disease mutation that you want to study, you do PCR to amplify it and get a bunch of that DNA. Any genetic trait that you want to study, do PCR, build up that section of DNA so that you can study
Yes,
it. It's
yes.
used for everything.
And it couldn't have this broad based applications unless it had the ease of use.
Yes. People would never sit there for 3 hours
Yes.
doing this constantly.
No, there,
Like,
there, there would be some dedicated people who would do this. It would be a
mhm.
very niche, what do you call it,
Or people would have built a machine or something maybe that would keep adding it every
adding maybe,
maybe,
but
but it
yeah,
it's
but
it's really a
the
pain.
technique, it would have been
Yeah.
a very niche technique.
Yeah.
It would not
It would have
be
been.
just widely used anywhere. Nowadays, you just have a thermocycling machine. You can go anywhere. You could take it to the fields. You take it here, take it there. It's a very
Yeah.
robust, robust application at this point.
Yes. You can
Any
use it on any gene, any piece of DNA.
anywhere, and
Yeah.
you can do so many things. You can manipulate so many different types of DNA from so many different situations, whether it's agriculture or forensic science or medical diagnostics, so many different widespread applications, just changing humanity, changing our lives for the better.
And I think people realize this because in 1989, Science magazine named Taq polymerase its first molecule of the year.
Yes, I can see that. It's I am trying to imagine, as we are talking, I'm trying to imagine something as revolutionary as PCR using Taq polymerase.
Yeah.
The kind of application that it has.
I think the next thing is going to be CRISPR. Like that's what people
Yes,
were like saying that CRISPR
yeah,
is like the next huge revolution in
yes.
genetic engineering
And
and
that
yeah.
also is based on PCR also. The
Yes.
foundation of that is
Yeah, so even
also.
that is like
That
it's
is,
not
yeah,
possible
it's
without
built on
this.
PCR. CRISPR,
Yes.
which is going to now revolutionize us again by allowing changes like CRISPR. Just a quick note
Yeah.
here, CRISPR is the technique that we can maybe employ to send a new piece of DNA into the body
Hmm.
and change characteristics. And people say in the future you will be able to change eye color, and you can be able to change hair color and all sorts of things for the person. If you already know that the baby has some sort of genetic disease, you can go in and fix that. They're already doing that using CRISPR. And so that's going to be the new revolution.
Hmm.
But even that, the foundation of that new upcoming technology is back to PCR.
And then PCR, of course, like we've been saying, would not have been possible without Taq polymerase and Taq Thermus aquaticus, which was discovered by Tom Brock and Hudson Freeze. So,
Yes.
thanks to its use in PCR, by 1990, there were over 1,000 papers citing the use of Taq polymerase. But
Yes.
meanwhile, Tom was continuing his research of thermophilic bacteria in Yellowstone. So all the
Yeah.
world is blowing up and he's just like, "Sweet,
I
I'm still in Yellowstone
yes,
studying these thermophilic
having the best
bacteria."
time of his life in a beautiful national park.
Yeah.
Good for him.
And so he starts looking for bacteria at even higher temperatures. And he does this by hanging slides on strings and then immersing the slides into the boiling waters of the geyser pools and then pulling them out a few hours later to check if anything's grown on them.
Yes, yes, yes.
And he wrote another paper called Life at High Temperatures, where he published evidence that there was bacteria that could
Oh,
grow at
wow.
above boiling temperatures.
Wow.
So that makes you maybe wonder, how hot can we go? So if it can grow at boiling temperatures where literally we thought no life can exist, what's the upper limit?
Yes.
And Tom thought that it would be possible to find bacterial life anywhere between 110 and 200 degrees Celsius,
Wow.
because 200 degrees Celsius is when the DNA nucleotides actually start to break down. So
Oh,
he's
wow.
like,
Okay, okay.
okay, so life
As long as
probably,
the, yeah,
as
as,
long as it's
okay.
DNA based, as long as the
Yeah.
life is DNA based.
Yeah, I don't know. I think as a microbiologist, I think the limit is 121 degree centigrade
Yes,
because,
you are
yes,
correct.
yeah,
Yeah. So in the 1970s, scientists
yeah,
discovered deep sea thermal vents, which brought renewed interest to his work.
yes.
In 1997, a team of scientists discovered Pyrolobus fumari, which
Okay.
is Latin for fire lobe of the chimney.
Ooh, ooh.
And it was discovered in a hydrothermal vent in the mid Atlantic ridge, and it
Oh,
grows best at 113 degrees Celsius.
wow.
But currently, you're correct, the most heat tolerant thermophile in general is the Geogemma barosi strain 121, which can grow at 121 degrees Celsius.
Yes,
And that's just in general. But the all
yes.
time record holder is the Methanopyrus candeleri, which can grow at
Oh,
122 degrees Celsius.
oh, wow. Oh,
So, right
wow.
now, that's our upper limits that we've just been able to discover.
Oh, wow. That's oh my gosh. 121 degrees centigrade, not Fahrenheit.
In Fahrenheit, that's like 250 degrees
Yeah,
Fahrenheit.
because yeah, no, 121 Fahrenheit is more like the very, very hot water that you would use at the most,
Yeah.
like a super hot water in which your plumber tells you don't do that, don't set, you'll burn yourself if you do that kind of thing. That's
Yes.
120 Fahrenheit, but 121 degrees centigrade is the limit.
Okay, so back to Tom Brock. In 1976, he started writing a book called Biology of Microorganisms, which was his first major textbook.
Hmm.
It did fairly well, with Tom continually revising it over the years.
Hmm.
In 1988, the fifth edition of this book came out, and it was the first full color microbiology textbook on the market, which, if I had been alive at that time and studying microbiology, that would have been a huge selling point for me.
You know, I think I've seen that textbook in my
Have
life as
you?
a microbiology student. Brock, yes.
Yes, that's kind of why I was wondering if you had heard of his name because,
I didn't make the connection. You know how I am with names.
yeah,
But
yeah, yeah.
once you told me the textbook and that color and everything, it
Yes.
clicked. Yes, I have seen the textbook. I
Yeah,
did have to
because this
study
would have
his
been in the
textbook.
80s. It would have been around
Yes,
this time when he's writing these textbooks and
correct.
you would have been
You're
going to
right.
school. Yeah.
You're right. And as I said, I'm not good with names, and so I don't make the connection. But yes, I don't
That
remember
I
the
mean that's better.
details.
Like I can't name a single author of any of the textbooks I've ever
No.
used. So, So his textbook is actually still being used today. It was
Yes.
renamed Brock's Biology of Microorganisms and is
Hmm.
currently in its 16th edition.
Yes. No, it was not named like that. His name was a small
Yeah,
underneath
I
it.
think it
The
was
name
renamed in
tag
like
was
2004 or something like
that makes
that.
sense. That
Yeah.
makes sense. Yes.
In 1971, Tom was offered the E.B. Fred Professor of Natural Sciences Emeritus position at the
Hmm.
University of Wisconsin in
Hmm.
Madison.
Hmm.
And
Hmm.
he was really excited by this because when he had been discharged from the Navy as a young man, UW had been one of his top school choices, but he had a
Nice.
hard time getting in because he was a non resident. He was living in Ohio at the time.
And now they are offering him a position.
Yeah, wonderful.
Isn't it beautiful how life has these little full circles that it
Yeah,
gives you? He's being offered a position in the university that he coveted as a
yes,
young man, and he's now being feted and honored and welcomed. Please come and
yes,
be one of us. And a
yeah,
position of power, not as a
yeah, as
lowly
a
graduate,
professor
lowly
now.
little student. Yeah, now you're a acclaimed, honored professor there.
Yes, please. You're not
Lovely.
applying to go.
Yeah,
You're
you're not
we're
applying. We
inviting
are
you.
honoring you. We are welcoming you. We are begging you to come to us. Wonderful.
Yes. Side note to the story, also around this time, his personal life changed quite a bit.
Hmm.
So at some point, he and Louise got a divorce, and I could
Oh
not
no.
for the life of me figure out why or what
Hmm.
happened. I did find it a bit surprising because she had been with him throughout all of his work at
Yeah.
Yellowstone and everything.
Yeah.
But for whatever reason, I guess it didn't work out. But he did get remarried to another woman named Catherine Middleton,
Okay.
not Kate Middleton, Catherine Middleton. Yeah.
Oh my God.
There's so many of these in this
Unbelievable.
story. Yeah.
Unbelievable fun names in this story.
Yes. She went by Kathy, and they stayed together for the rest of his life. Officially, his project at Yellowstone ended in 1975, but afterwards he started traveling the world looking for exotic microbes. He
Hmm.
developed into quite the travel bug, no pun intended.
Yes.
He was visiting hot springs in Iceland, Japan, the Caribbean, New Zealand, Italy, and Central America.
Good
And
excuse to go to all these
yeah,
places.
why
Hmm.
not?
Why not?
Yeah, and he found hypothermophiles in pretty much every hot spring he visited.
Mm-hmm.
But he
Hmm.
also kept going back to Yellowstone. He simply said because it was the most accessible location where
Hmm.
so many microbial habitats existed.
Yes.
But I'm sure it was also just a special place in his
Yes,
heart at this
yes.
time.
The place where he was initially so unwilling to go became so close to his heart.
Yeah.
That is again ironic. There is so many little circles of life ironies in this
Yeah.
story.
But he also started turning his focus a little closer to home by looking at the ecosystems around the Great Lakes since he's now living in Madison.
Yes.
And he felt like he should give back to the taxpayers of Wisconsin who are essentially funding all his research.
Okay. Okay.
So in 1981, he helped found the North Temperate Lakes Long Term Ecological Research Network to
Hmm.
fund long term limnology. This is a new term I hadn't heard of.
Yeah.
Limnology is lake research.
Oh,
Yeah,
nice. I did not know this. Okay.
yeah. Also, around this time, he becomes once again interested in the history of microbiology. And so
Hmm.
he starts, going back to the kind of like he had written a textbook called Milestones in Microbiology. And he once
Hmm.
again comes back to
Hmm.
updating that. And
Yeah.
he was surprised to find that there wasn't really a good biography of Robert Koch, the original microbiologist, German microbiologist.
That's the one whom he translated initially. Yeah.
Yeah. There wasn't a good biography of him in
Yeah.
English. So once again, he uses his German translating skills to write one. And I was like,
Ah.
Thank you for writing that because I will probably use that in a future
Yes.
episode on Robert
Yeah,
Koch,
and it's
you
the
know?
writer in him coming out. Initially
Yeah.
he wanted to be a writer. And he has that science communicator, educator feeling in him
Yes.
all the time. So all of it is coming out to create these beautiful pieces of work.
Yeah, and I love that
Nice.
he thought it was important that Robert Koch's biography gets told. So not like he had spent time already obviously translating his research and his scientific work, but he's like
Yes.
there's no biography of this guy's life and
Yes.
I was like, "Yes, that's
Yes.
exactly why I have this podcast too."
That's right. We need to understand where these scientists are coming from. This is
Yeah.
it's amazing.
He and his wife, Kathy, also started an academic publishing company called Science Tech Publishers that they operated for 12 years. Overall, during his career, he published over 250 papers and 20 books.
Wow.
So
Lot
he
of
got
work.
that writing in
Yes.
anyway. Doesn't matter.
Yes. Yes. When I say,
Hmm.
who am I? When I ask the universe, who am I? And something comes to you, you
Hmm.
know. And if you're doing something else, then somehow you will find your way back to the core of who you are.
Yes.
So it sounds like at the core, he's a writer, he's a creator, he's a very inquisitive guy who looks around him, sees things that are different, and then writes about it, educates others about it. That's
Hmm.
at the core of him. That's who
Yeah.
he is.
Yeah.
And it doesn't matter what setting you put him in.
Yeah.
That core identity of him comes out.
Yes. So he retired from the University of Madison in 1990 at around 65 years old, but he was far from done from working. With all of the royalties that Tom and Kathy had from their years in publishing, they put that all to good use. During his work, kind of understanding ecology and specifically the unique environment in Wisconsin, Tom learned about an extremely rare kind of ecosystem that existed called an oak savanna.
Hmm.
These oak trees thrive
Hmm.
in dry areas with poor soil quality, and very interestingly, they depend on fairly frequent fires to survive.
Oh, wow.
So the oak tree themselves are fairly fire tolerant, but if there's any like invasive species like buckthorn or honeysuckle that's growing in the area, they get burned away and allow the oak tree to thrive.
Wow. Again, it's an oxymoron. It's like you would think the oak tree would not survive the fire, but it's opposite.
Yeah,
That's
they've
interesting.
developed or they've evolved to be able to survive and thrive in these types of extreme environments again. Like this
Yes.
fire is raging and they're like, okay.
And all the ash serves as fantastic fertilizer for the oak tree to thrive even better.
Yeah. And this is a very rare ecosystem that exists in North America and a couple of other places, but it does exist naturally in Wisconsin.
Hmm. Hmm.
Um, but when people like settlers came to Wisconsin and they started farming the land and settling in the area, they stopped letting any fires burn through the area. And so now these oak trees are essentially getting overgrown and choked out and dying due to all
Mm.
of these invasive
They
species.
need the fire to survive well.
Yes. So Tom and Kathy, with all of their money now, bought 140 acres of abandoned farmland about 20 miles west of UW's campus and established the Pleasant Valley Conservancy. And they slowly started restoring the land back to its original state,
Hmm.
encouraging the original biodiversity that was part of the
Oh,
ecosystem to return to the area through controlled burns, basically.
that is a very good legacy. They really are giving back, respecting that nature and giving back and making sure
Hmm.
that the nature that they are so in love with is protected.
Yes. They
Wow.
developed a lot of restoration procedures that became very useful for land and conservation in other wildlife preserves and national parks. And today you can go and visit Pleasant Valley Conservancy throughout the year if you want to observe the wildlife that has returned to the area.
Nice.
Over the years, Tom and Kathy received numerous awards for their efforts in conservation and restoration. And of course, ever the writer, he published 2 books about their restoration efforts.
Everything he comes across, he writes,
Yeah,
catalogs. Very nice.
which is part of the reason why this episode is so long and detailed, because I had so
Yes.
much information to
Yes.
draw on for this
But
episode, which
that's
was
that's important
fantastic.
because it's
Yeah.
it's it's a nice learning experience for all of us.
Yes.
We see where they have been and their efforts, and we respect that, move forward, build on that legacy.
Yeah. And he saw the importance of sharing not just his work, but like how he got there, his life,
Yeah,
and, everything that led to these discoveries.
truly wonderful.
In 2019, he received an honorary degree in microbiology from the University of Wisconsin to acknowledge
Hmm.
his extraordinary lifetime accomplishments. In 2021, Tom died after taking a fall in his home in Madison. He
Hmm.
was 94 years old. So,
Okay. Okay.
very
Yeah.
long, good life.
Good productive life, a good legacy.
Yes.
And sometimes it's not necessary. The fall is just the ultimate
Yeah.
cause. It's not, it was old age and he had to go
Yes.
one way or the other. That is so. But he
Yeah.
had a good life. He had a good
Yes.
run there. And
He
he
had a
did
really
good
good
with the
one.
life that he was given.
Yeah. I mean, changed
Wonderful.
the world.
What a beautiful story.
I had known that Thermus aquaticus had been discovered in Yellowstone, and that's kind of why I went chasing his story. But
Yes.
I love
But
how he was so obsessed with extreme organisms and how
yes.
all these, like, even up to an oak tree, it's not just microbes, but it's like,
Yes.
oh, this oak tree that lives in fire. I want to learn about that. That's an
But
extreme
there is
organism.
so many lessons there in terms of even like, okay, these oak trees, how do they survive? For example, okay, fire is very dramatic, but
Yeah.
they seem to survive in poor soil with not as strong with nutrition.
Mm-hmm.
How do they do that? Can we
Yes.
apply that to other
And we learn from that.
organisms?
And yeah.
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 Can we help other trees to grow where the soil characteristics have depleted and so they are not able to do so well anymore?
Yeah.
So many good lessons.
It's going to be huge. It would be huge for agriculture and stuff because, whenever you grow crops, they take up all the nutrients from the soil and then the
Yes.
soil gets depleted. And
Yes.
then we have to add that back by
Yes.
either using fertilizer or we have to rotate crops so that we're not using the
Yes.
same land every single time, every single
Yeah.
year,
Yeah.
which of course means you require a ton more land than normal. But if you could maybe in like using PCR and using
Yes.
CRISPR and all these things, if you could insert
Figure
a
out.
gene
Yeah. Yeah.
into
That allows them to
that
thrive
allows them to,
anyway.
yeah. And
Yeah.
especially with climate change and all these things happening, the
Yes.
soil is getting worse because of the dry heat and,
Yeah.
lack
And
of rain
and
or
temperatures
whatever.
are going to higher levels than they were before.
Yeah.
So how do we survive that? These extremophiles would probably give us a few lessons in that.
I mean, it's kind of sounding sci-fi-ish, but 20 years ago, CRISPR sounded sci-fi-ish. So
Yes.
you never know.
Yes.
You never know
You
where
never
science is
know.
going to
Yeah.
take you. So
Yes. Yes.
yeah.
And how important these findings are as we move forward.
Yep. Thanks for listening. If you have a suggestion for a story we should cover or thoughts you want to share about an episode, reach out to us at smarttpodcast.com. You can follow us on Instagram, TikTok, and Blue Sky at Smart Tea Podcast and listen to us on Spotify, Apple Podcasts, YouTube, or wherever you get your podcasts. And leave us a rating or comment. It helps us grow. New episodes are released every other Wednesday. See you next time.